Alrighty, folks. Good afternoon, good evening, good night, or good morning. This is Dr. Kaz, and I am doing another Blackboard Collaborate review session, optional, of course. And I'm going to be going over Chapter 4, all right? And I probably won't get through all of it in this session, but that's okay. I'm going to try to get another one out there before we have our Collaborate session on Sunday, this upcoming Sunday. September 13th at 7 p.m. Anyways, let's jump right into things here. Let's talk about the biology of a cell. And one of the key points that I want to highlight for you folks is that cells, as I talked about earlier in Chapter 1, we're going to go through the organization structure, all right, of an organism, that being us, the human body, in this course. So we are going to start off with the smallest structure, which was the atoms and molecules, and then we're going to build there. So the next structure that we're going to move on to is the cell. And you need to know this. The cell is the functional unit of the body, okay? All these body processes that we're going to be learning about, the general concepts that you're going to learn about, you know, digestion, blood flow, heart rate, all that is dependent upon these cells and the activities that these cells actually perform. So if you understand how a cell performs and you understand its structure, so both the physiology and the anatomy of a cell, you're going to understand a lot of the crucial concepts in this course much better, okay? So we're going to start off by talking about the three common features, all right, or the big three, as I like to call them, components of a cell. All right, we have the plasma membrane, the nucleus, and the cytoplasm. Okay, so we're going to start off, all right, on this slide by talking about the plasma membrane, but when I get into the detailed aspects of the cell, I'm going to start off by talking about the cytoplasm, and then we'll spend a lot of time talking about the plasma membrane, and then I'm going to finish up with the nucleus. All right, so the plasma membrane is the envelope that essentially surrounds a cell, and it keeps everything on the inside of the cell inside the cell, and it keeps everything on the outside of the cell outside the cell. So it's going to separate the internal environment of the cell from the external environment. Important concept that you want to know is that it forms a limiting barrier. Okay, so it's going to limit what moves in and out of the cell, and we'll get into that later on when we talk about the plasma membrane. Okay, some of these plasma membranes have these modifications to them. All right, and we've talked a little bit, or sorry, I keep confusing you with my live classes. You probably looked that over, all right, when you were going over the tissues in lab. Okay, but we're going to get into more detail about them. We have our cilia, our flagellum, and the microvilli. And we'll discuss those. Okay, cilia are the hair-like projections, all right, that come off of the apical region of a cell. That's the top part. And it moves things by the cell. So the cell will stay stationary, but the whatever the cilia is moving will move past the cell. The flagellum is much larger than the cilia. Okay, it's like a little tail. All right, bigger, thicker, longer. All right, and it propels the cell. It moves the cell. All right, so there's only one cell in the human body that has a flagellum, and that's the human sperm. Okay, no other cell has a flagellum except for the human sperm. You should know that because that gets I've seen that asked on tests. Then finally, we have our microvilli, or as we've seen when we were studying the tissues, the brush border. And the microvilli, they're essentially needed for increased surface area, so we can maximize the amount of absorption that occurs. For example, in the digestive tract, all right, you have these villi, these structures, these projections that come off of the small intestine, all right, and those villi are lined with these small epithelial cells, all right, the simple squamous the ciliated simple columnar epithelium, all right, and they have microvilli there to increase the surface area for digestion. Okay, all right. The next is the nucleus. That's the control center for a cell. Okay, it's the largest visible structure through a microscope or an electron micrograph or whatever you're using, all right, that's inside the cell, right? And much like the cell has a plasma membrane, well, the nucleus has what we call the nuclear envelope, right? And that separates the internal environment of the nucleus from the cytoplasm, okay? But inside the nucleus is where we find our DNA, our genetic code, genetic material, chromosomes. All of that is stored inside the nucleus there, okay? And much like a cell, all right, with cytoplasm, the nucleus has its version, all right, of the goo inside, and that's called the nuclear plasm, okay? Then the third component of the cell is the cytoplasm, all right? That's the structure that occupies, all right, the space in between the plasma membrane and the nucleus, okay? And there's three components to it, and we'll talk about each of these separately: the cytosol, the organelles, and the inclusions, all right? But this is where a lot of cellular activities, metabolic functions will occur. So we'll start to talk about that. We'll talk about all the different aspects of the cytoplasm. All right, so let's start off by talking about the cytoplasm. All right, there's three parts to it. First part is the cytosol. All right, another name for that, and you want to be familiar with it, is the intracellular fluid. Intra stands for inside. So this is the fluid inside the cell, because you also have extracellular fluid, all right? And that's the fluid outside the cell, okay? So this type of fluid, all right, it has a lot of water in it, but it doesn't have that water consistency. It's a little bit thicker than that, kind of like motor oil, all right? It's going to be a little bit more viscous, okay? So, of course, due to the high water content there, you are going to have dissolved substances in the cytosol, namely macromolecules. And we learned what the macromolecules were: proteins, lipids, all right? Not so much lipids because they won't really dissolve, but sugars, all right? And different ions. And that'll come into play later on when we learn about, like, the neurons. You have a high content of potassium inside the cell, okay? Okay, the next structure or component of the cytoplasm are the organelles, which mean little organs. So much like us as humans, we have organs throughout our body: heart, liver, lungs, all right, stomach. Okay. Then they all perform functions that are crucial to our needs, all right? The organelles inside the cell do the same thing for the cell itself, okay? So they have a variety of functions which we'll get into. But there are two types. The first one are membrane-bound organelles. And that means these organelles have a little membrane, right, that goes around them. And so because whatever function is, and in some situations, all right, these organelles need to separate their internal environment from the external environment or the cytoplasm, because they might be digesting something, and they don't want the digested contents that's inside the organelle to spill out into the cytoplasm, because it could damage the cell. So we put a little bit of an envelope around it, like a bubble, okay? So we can separate the two. And then the other type of organelles, those are going to be what we call the non-membrane organelles, okay? So we're going to talk, all right, about these organelles, but before we do, all right, I want to kind of go into a little bit more detail about membrane-bound organelles and non-membrane-bound organelles. All right, like I said before, the membrane-bound organelles have a membrane, so we can separate whatever's happening inside the organelle from the cytosol. And you can see the list here of what it includes, and we'll go through all of those individually. Okay, I won't spend too much detail on them, but I do want to go through a few of them just to kind of give you a decent understanding. The other type of organelle, like I said, is the non-membrane, and those do not have a membrane, okay? But these organelles are composed of protein. You remember all the different functions of proteins: transport, catalyst, defense, all right? So a lot of these membrane-bound organelles are going to carry out some of those functions that we talked about for proteins. And you can see a list here, right, of the non-membrane-bound organelles, which we'll get into here. All right, and the final part of the cytoplasm are the inclusions. The big thing that you want to know about inclusions is that they're just temporary. They don't stay very long, okay? So they come and go. And a lot of the time, these inclusions are just these molecules that may be needed for some sort of chemical or metabolic process, all right? Or they could be left over from a metabolic process, or they could be a damaged structure. Regardless, all right, they just kind of float around haphazardly in the cytoplasm. So you can see here some of the examples when we were talking about glycogen here and how the liver and your skeletal muscles will create glycogen, and it stores the glycogen in those tissues for a rainy day, right? It stores it directly into the cytoplasm there, right? Adipocytes will store triglycerides. All right, so like I said, these are temporary. They come and go based on the needs of that cell. All right, so here you can see a picture, all right, this is in chapter 4, 4.4 figure. All right, and you can see some of these structures here. Here is the plasma membrane, and you can see the different plasma membrane extensions. Here are your microvilli, pretty uniform in shape, okay? Then you've got these hair-like projections. Those are the cilia. You got this real long and thicker projection coming off of the plasma membrane, and that's the flagellum. That's the one that will move the entire cell. You can see the plasma membrane encloses itself around the entire cell, and then you can also notice the nucleus here. Okay, and it has its own version of a plasma membrane, which we call the nuclear envelope. And all this stuff in here is the chromosomes. All right, or excuse me, are the chromosomes. That's your DNA, your genetic information. And then you can see in the cytoplasm, we have a variety of different organelles, which we'll talk about. Okay. All right. So a couple functions, and it's crucial that you understand these functions of a cell. Just a few things to try to draw your attention to. Okay. Cells perform several different functions, but at all times they always have to maintain the shape that they have, like a squamous cell, a cuboidal cell, a columnar cell, because a lot of those shapes are function dependent. All right, so not only do they have to maintain their shape, they have to maintain their integrity. And what that means is, all right, integrity is like the support, how strong the cell is. So all this has to do with, all right, how strong, all right, your plasma membrane is, and then also the internal contents of the cell, all right, whether or not they are functioning appropriately. Okay, so hey Tina, this is one of those things that you need to understand when we're discussing, all right, some of the functions of cells. All right, so much like us, when we're performing certain metabolic functions, like when you eat an apple and you digest the food and you use that food, that energy from an apple to perform certain metabolic functions, your cell does the same thing. So what it has to do, it has to take nutrients into itself from the external environment. Okay, so it has to obtain these nutrients, and then if it's a cell that needs to build like a hormone, let's say, for example, insulin. Okay, beta cells in the pancreas, all right, are going to generate and build insulin. All right, so it'll need some of these components here to build that. So that needs building blocks. So amino acids to build proteins and proteins to build insulin. Okay, amongst other things. Just like with us, when you eat food, you know, maybe later that night or the next day, you have to go to the bathroom. What are you doing when you're going to the bathroom? You're getting rid of wastes. Well, the same thing that a cell has to do. Okay, it's going to get rid of its wastes, because if it doesn't, those wastes can accumulate inside the cytoplasm and keep building up and keep building up. And if the cell cannot get rid of those wastes, then it can cause damage to the organelles inside, or it can prevent those organelles from conducting their normal metabolic activities. And if that happens, the cell could also die. Okay, so it has to get rid of them. Some cells divide in your body, and some cells don't. All right, the majority of cells do divide. All right, and they'll divide either through mitosis or meiosis. Mitosis is what we call somatic cell division. So all the cells of the body, all right, that can divide will divide by mitosis. The only cells that divide through the process of meiosis are your sex cells. So that includes the eggs in females and sperm in males. Okay? So cells that don't divide, we call those amitotic. Right? And the purpose of cell division will be, one, if you're growing, all right, you need more bone to grow taller, all right? Or if you damage something, okay, if you've fallen and scraped yourself or damaged, a bone, like if you broke your bone, you need to repair it. All right, and as we get older, we need to replace older cells with newer cells, okay? So let's start off by talking about some of these organelles inside of our body, okay? The first one here, we're going to start off with the membrane-bound organelles, all right? The first one is going to be the endoplasmic reticulum, okay? And you can see here, our endoplasmic reticulum is this tubular structure, all right, that surrounds or is in close proximity to the nucleus, okay? And there's two types of the endoplasmic reticulum. One is referred to as the rough endoplasmic reticulum, and we call it rough because it's got these little bumps on it. And those bumps are called ribosomes. And ribosomes, all right, will actually make and build proteins. That's their job. Now, on this other type of endoplasmic reticulum, you don't see the ribosomes. All right, so it's essentially smooth. So we refer to that type of endoplasmic reticulum as smooth endoplasmic reticulum. So you can see an electron micrograph showing you here, all right, you've got some free ribosomes just kind of floating around, and then you'll have some ribosomes along these tube-like structures, and that's the rough endoplasmic reticulum, okay? So the rough endoplasmic reticulum, its main job is going to be, or one of its main functions is to make proteins that it's going to secrete to one of two places, okay? Well, let me rephrase that. Not one of two places, but it's going to make proteins that are going to be sent to either the plasma membrane, okay? And say the plasma membrane needs to repair something or something's old, all right? It'll send proteins into the plasma membrane. It can also send proteins out to be sent outside the cell, okay? But it also is going to make, all right, proteins that are going to act as enzymes, those catalysts, for another membrane-bound structure called the lysosome. I don't know if I have a picture of it. I don't. All right, we'll talk about what a lysosome does, all right, in a few slides here. Okay, so that's what it makes. At the same time, it's going to modify certain proteins, okay? We've got our glycoproteins, which are highly prevalent in ground substance. So, for example, you can make a glycoprotein, right, and simply by adding a carbohydrate group to that, all right? And it also store some of our proteins there, okay? And one other thing that I want to draw your attention to is this guy right here, the peroxisome, all right? The peroxisome also gets made here, all right, in the rough endoplasmic reticulum. And we'll talk about the role of a peroxisome shortly. All right, but again, they play a pretty big role in digestion, also in beta oxidation, which is breakdown of fats and trying to get energy from that, okay? The other type of endoplasmic reticulum is going to be our smooth endoplasmic reticulum. Smooth endoplasmic reticulum are going to be found in organs that detoxify drugs, like your liver, for example. All right, it detoxifies drugs, alcohols, certain poisons that you might take into. So it pretty much is like a filtering center. At the same time, while it's detoxifying, it also is able, all right, to make certain steroids, right? So steroids are a hormone, all right, that your body, all right, I won't say requires, but will make, especially in males, all right, for like testosterone, for example, okay? And then finally, when we undergo that liver, I'm telling you, when the liver has all that stored glycogen in it, and it's time, all right, to make more, all right, or break it down, but make more, all right, it will be made there also in, all right, the smooth endoplasmic reticulum. All right. The next structure I want to talk to you about is one of my favorites. This is the Golgi apparatus. Whoops, hold on one second. Let me zoom in. All right, so this is what the Golgi apparatus looks like on an electron microscope here, okay? But slide down here to kind of show you. Think of the Golgi apparatus as the warehouse of the cell. And what it does here is it will modify certain proteins, okay? And it'll package up certain proteins, okay? So two types of proteins that are going to pretty much go to the the Golgi apparatus, all right, will come from the rough endoplasmic reticulum. So before I forget, I needed to mention that the Golgi apparatus is what we refer to as a polar structure, okay? It doesn't mean it's positive and negative, but what that means is the end that's closest to the nucleus or the endoplasmic reticulum, we refer to that as the cis phase. That's where these proteins will enter into. And then the end that's furthest from the nucleus or closest to the plasma membrane, that's the trans phase, okay? So what will happen is that the rough endoplasmic reticulum packages up these proteins, makes a secondary vesicle, sends it over here, all right, to the cis phase of the Golgi apparatus. The Golgi apparatus will then modify it, okay, and send it through a couple of stages here, add some things, take some things away, all right, and then it'll package it up, and it'll send it to one of two places. Hey, Rowan. It'll send it either to the plasma membrane, all right, and of course, in order to send it to the plasma membrane, it packages it up into what we call the secretory vesicle. It gets sent over here to the plasma membrane, and then it'll combine with the plasma membrane, and the proteins that were inside of that secretory vesicle will be added to your plasma membrane. The other one here, all right, will be secreted from the cell itself. So those proteins that are made for that will get packaged up again in a secretory vesicle and then sent to the plasma membrane. Same thing as before, it'll merge with the plasma membrane, but instead these proteins will be excreted out into our interstitial fluid, or what's also called the extracellular space. Okay? So that's pretty much, that's the big role. Oh, and one other thing. The other part, excuse me, is it'll also make proteins, all right, that are going to be incorporated into the lysosome. All right, the lysosome is another membrane-bound organelle, okay? And essentially its job is to go around the cell and kind of scoop things up, all right, that were maybe half made, all right, and it will digest these unneeded substances, okay? So, for example, okay, you can see, all right, through this process here, exocytosis, where the cell is getting rid of some of this protein. If some of it doesn't get excreted completely outside the cell, it might float around here, okay? So this lysosome will just go around like a Pac-Man and gobble it up. So what the Golgi apparatus does is it helps to make these digestive enzymes for the lysosome. In chapter 17 in Bio 211, you're going to learn that the lysosome plays a huge role in thyroid hormone synthesis. Because essentially what happens is that the thyroxine is inactive. It will be inside of a thyroid cell, and lysosomes will then take in the thyroxine, and the digestive enzymes inside that lysosome will cut things away from the thyroxine molecule and take it from its inactive form into its active form. And then the cell will excrete thyroxine into your blood, and then it goes to wherever it needs to go. Okay? So lysosomes play a role in the actual activation of certain structures too. Okay, so here is our lysosome. Okay, and mainly it gets all of its digestive enzymes from the Golgi apparatus here. And what they do is they just wander around the cell, all right, and they clean up unneeded structures, damaged proteins, damaged organelles, things that are just kind of lying around here, all right? All right. Not proteosomes, peroxisomes. Again, you'll find out that these membrane-bound organelles, the cell itself has all these little organelles that play a role in kind of keeping things clean, I guess is the best word for me to describe. Basically, these organelles will float around the inside of the cell, and kind of much like if you've ever gone to a parking lot for like a mall at 3 o'clock in the morning, I don't know why you'd be there, but you'll see these like huge vacuum cleaner like trucks driving around and cleaning up the trash. Where your cell has structures that do the same thing. All right, the lysosome is one of those. We'll talk about the proteosome in a few minutes. Well, maybe probably not this slide, but peroxisomes, again, they do something similar. All right, what they'll do is they'll wander around and they'll break down certain structures, all right, fatty acids during the process of beta oxidation, all right. Also, they help to make lipids, all right. Also, when they're wandering around the cell, they will go around and kind of clean up certain leftover products also. But something that I find really interesting about these organelles is that they make this stuff right here, H2O2, which is hydrogen peroxide. And see, when you're not using it to dye your hair, all right, or clean acne off your skin, off your face, your cells use it to help kill bacteria or viruses. They're very, very effective that way, okay? All right, one of my favorites, the mitochondria. This structure is also nicknamed the powerhouse of the cell, because it's this structure here that generates a lot of energy, okay? And if you remember what the energy currency, all right, of a cell is, it's ATP, all right? But mitochondria need one thing to be effective, all right, to actually make, all right, a lot of ATP. And that one thing is, I just wanted to pause there for some of you to think of what it is, oxygen, okay? Oxygen. Because with oxygen, you can engage in what's referred to as aerobic cellular respiration. If you do not have oxygen, we cannot get, all right, ATP made effectively. Let me rephrase that. One glucose molecule, right? Without oxygen, one glucose molecule will only yield you 2 ATP. It's not bad, but it's not very good. If you have oxygen present, that same single glucose molecule can yield you 32 ATP. So essentially, you need oxygen to get into the plasma membrane. It's like that guy that knows somebody that can get you into a bar or a club. That's what oxygen does. And it'll get one of the products of glucose into the mitochondria so you can break it down into 32 ATP. Okay? So that's its main function. Mitochondria, all right, is to generate aerobically, through aerobic cellular respiration, 32 ATP. All right? It also does contain, all right, genetic information. And I talked about that before. I think I talked about that with you guys. Maybe not. But you do have, all right, 16 DNA strands in your mitochondria. All right, ribosomes. Those are those little bumps that I was telling you about. All right, these guys are non-membrane-bound organelles. Okay, so they do not have a membrane around them. The only thing for this class, what I want you to know about a ribosome, I'm not too concerned about a large subunit and a small subunit, okay? But what ribosomes do is making protein right here, protein synthesis, okay? So the ribosomes that are bound to the endoplasmic reticulum, excuse me, are going to make proteins that are going to go to the plasma membrane or be exported from the cell or go into a lysosome. The free ribosomes, that's these guys that are just floating around, they're going to make the proteins that are going to stay inside the cell. They don't leave the cell. Okay, the only proteins that have the possibility of leaving the cell are the ones that are bound here onto the rough endoplasmic reticulum. Okay? All right. All right. Our next non-membrane bound organelle is the centrosome. Okay? Up until recently, there wasn't a lot known to the function of the centrosome except during cell division. Okay? So these two structures, you don't really even see them, all right, in a cell. They don't become very visible until it's time, all right, for a cell to divide. Okay? So their big role, all right, is through cellular division, all right, when essentially what we'll see here is, all right, you got your cell here, and then normally they stay in this configuration here, all right, perpendicular to one another, okay? But when it's time for the cell to divide, what they'll do is one will go to one end of the cell, the other will go to the other end of the cell, and then they create what's called the mitotic spindle, all right? And these microtubules that extend and they go right to the middle of the cell. And essentially what they do is they pull the cell apart from one another, okay? And they'll split the cell right down the middle. So now you'll end up with two cells where you once had one cell, okay? Now, one of the other functions that they do that they discovered recently is that they help to organize these microtubules. Those are these proteins that we use, and I don't know if I have it in this slideshow or not, but when you were, but when you were reading, if you're not sure, go ahead and look into your textbook. When you were reading about the cytoskeleton, all right, microtubules are one of the structures that protein structures that are utilized for the cytoskeleton. All right, but the centrosomes will help in the organization of these microtubules, so it can help to what's the word I want to use? Contribute to your cellular support structure. Okay. All right. One of my favorites, and I know I keep saying that for a lot of these cells or organelles, but I love proteosomes because proteosomes are the quality control expert inside your cell. A lot of times when you're making proteins, things don't go as planned. It just happens. For any of you that have worked in production, like say for BMW, not every car that comes off that line is perfect. All right. Same thing with protein. When the ribosomes are making the protein, they might make a mistake. Something might happen. And so you get an imperfect protein. So what your body does, or excuse me, what your cells do is that they have this protein that goes around. It's called ubiquitin. And it gets added on to an imperfect protein or a damaged protein, or even a protein that's just ready. It's an old protein, and your body's great at recycling, so it wants to use the amino acids in that protein to make maybe another protein. So it tags it with this ubiquitin. So then you got our proteosome, our quality control expert. It's floating around there, and it sees the ubiquitin tag on this protein, all right, that may be damaged or half-made or whatever. And what it does is, it's like a string of pearls here. It just shoves it down through the organelle. And as it's getting shoved down through the organelle, it breaks it apart back down to its functional units. And as you know, the functional units are amino acids. All right. And in some cases, it doesn't break every single one off individually, and that's okay too. But then, if you remember from chapter 2, okay, if you have 3 to 20 amino acids strung together, then that's a peptide, okay, or peptide fragment, okay? All right, so you can kind of see that this is great for quality control, and a lot of times, because very rarely do we want to, we never want to have any type of improperly made protein or anything, because then that can lead to abnormalities and pathologies and whatnot. And so this course is about when things are normal. You want to learn about the abnormalities, you take a pathophysiology class. You'll learn it real fast. Oh, good. I did include the cytoskeleton into this lecture. All right, so the cytoskeleton. Important thing that you want to understand, just like we have a skeleton in our bodies, our cells have a skeleton. All right, the bones in our body help to support us, while the cytoskeleton helps to offer the cell internal support. But at the same time, the cytoskeleton allows us to organize, all right, where certain organelles should be. You don't want them all clumping up in one area, all right, or gathering to another area. When I was talking about the centrosomes, during the creation of the mitotic spindle, when it splits apart, all right, we use our centrosomes to extend those microtubules out there that help to pull the actual cells away from each other. And then when we were talking about the exporting of those proteins in those secretory vesicles, well, those secretory vesicles aren't just floating around randomly, all right? You have these microtubules that are built, all right, kind of like a railroad track, all right, that go from point A to point B, and the secretory vesicle kind of rides on that railroad track like railroad cars do on a train. Same type of scenario there. Okay, so you can see that cytoskeletons play a big role, all right, in a lot of the functions inside the cell. So there's three types: the microfilaments, the intermediate filaments, and the microtubules. That's as much as I'm going to go on that for here. If you want to get into a little bit more detail, go ahead and check it out in the book. All right, and then the last thing that I want to talk about in this video is going to be the cell's external surface. I mentioned it before about the three modifications that you'll see: cilia, flagella, and the microvilli. The first two, all right, I want you to think movement. All right, cilia and the flagella. Think movement. Whereas the cilia, those cells stay stationary, and the cilia, these hair-like projections, kind of whip back and forth and move things across the surface of the cell. All right, so like in the fallopian tubes or the uterine tubes, all right, with the oocyte, the egg, when it is ejected out of the ovary, all right, the cilia beat back and forth and sweep the egg down into the uterus, okay? And the mucous membranes of your respiratory tract, all right, the cilia beat back and forth to keep the mucus from going down into your lungs. It wants to beat it up into your throat, okay? Only cell that contains the flagella, I mentioned this before, is the sperm cell. Okay, and that's the structure. It's much bigger than the cilia that will move the entire cell. And then finally, the microvilli, we've already talked about that before too. It helps to increase the surface area of the cell for the proper or the maximum amount of absorption that we can get. We'll see microvilli a lot in the digestive tract. All right, I'm going to conclude this again. This is going to be a short video, and it's because I got to go and take my daughter to karate. And I don't want to overwhelm you with too much material. I will put this up very soon. Obviously, if you're watching this, I've already put it up. So make sure that you get a chance to review this as much as you'd like. And I will try to get out the rest of Chapter 4 before our Blackboard Collaborate session on Sunday. All right, I hope you enjoyed this video, and keep studying. If you have any questions, email me.